Welcome everybody. It's time today for a new session of the COMPAMED High Tech Forum virtual. So it's a very pleasure for me to be here today. Francesca Moglia is my name and I work for EPIC. And today we have a special EPIC TechWatch about laser and photonics applications. So today I'm really happy to be here. I have to admit, I would have preferred to be in Düsseldorf, but you know, now we are getting used to this online world, and it's very nice that COMPAMED still can take place completely and fully virtually so that we can still meet, make business, and talk about technology. So now I will share my screen briefly. I see many familiar faces here. So many EPIC friends are here today, so I know that I don't need to make a super large EPIC introduction, but still, I know maybe in the audience and also on YouTube, we have someone that doesn't know EPIC, so please let me spend a couple of words about it. So now, EPIC, easy. So since a few weeks, we can still say, we can newly say that EPIC is the largest photonics industry association in the world. So it's really nice to show this slide. You see now the logos of our companies, they are more than 620. They are getting always smaller, but I promise you that you see them also here behind me, always very small, but some of our speakers will have for sure a few larger ones on their slides later on. And also you can see all of them, of course, on our website. So to know exactly who is the photonics ecosystem that we have in our members. Now the next question is, who is EPIC? And actually the answer is still the members. So these are still the 620 plus companies, this guy behind me, the slide before, but nevertheless, there are some few people now, a team that sometimes is on stage like today, but most of the time is behind the scene. These 14 amazing people you see here in this slide. And these are those that are helping our members to be basically in collaboration among each other with some technical support to interact also with end user outside the consortium. And basically half of the team has a technical background in photonics. The other half is specialized more in event management and marketing, and also in investment. And so we have covering all the aspects that for business interactions is really important. So what we do, there is a few information on this slide. So we organize events, we organize networking opportunity, we take care of investment, especially our CEO, Carlos. We offer now mentoring programs as well. We have a full page of jobsinphotonics.com, really, where all our members can put all their open positions. So it's really nice to have a collection of this. We have market data. So for our members, we have market reports. And since basically the pandemic started, we have a full portfolio of products that is online, and since after summer, we have, for example, virtual company visits where we really come to your company virtually. We have a virtual, so online product launches and product releases, but also, and that is something that we started in 1st of April already, that it sounded like a joke at the beginning, 1st of April, but then it became very soon very serious. So now we did 50 of these events. They are called EPIC Online Technology Meetings. And basically they are 2 hours online with the full value chain of a topic. People that are discussing. Some have a very short pitch talk where they explain basically opportunities of collaborations with all the participants in the audience, and also lively discussions. So that's what we do, and that's what we did also. You see here, the gray part is getting larger because this is the program of fall, so till Christmas, but don't be worried. For sure, we will very soon now publish the new program for Q1 of 2021. But now let me focus a bit for the audience of today, and also for the speakers that we have in the room today. Is probably very nice to know that we have 2 upcoming events till December, so till Christmas actually, that could be really interesting for you. So Photonics for Improved Pharma Processes and Photonics for Medical Devices, these 2 red in the box, and 30th of November and 9th of December are the dates to be saved. So hopefully we will see you also in these 2 occasions. But now I don't want to steal more time to our great speakers today. So as you see here, we have 8 speakers today. This is the first part, second part till 1:20. So now it's a very big pleasure for me to introduce Ralf Liedert, our first speaker from VTT, Technical Research Center of Finland, that today actually is in the best position of presenting one of our beloved pilot lines, MEDPHAB. So the Europe's first photonics pilot line for medical application. And so now I stop sharing and I leave the floor to Ralph. Thanks a lot.
Thank you so much. Thanks so much. All right. Hello, everybody. My name is Ralph Liedert from VTT Technical Research Center of Finland, from the cold and north and rainy Finland at the moment. I'm going to talk to you today a little bit about MEDPHAB, and I'm sharing my screen. And there we go. So what is MEDPHAB? We have received funding from the European Union to establish a European pilot line for photonics devices, medical photonics devices, which is very, very unique. It's the first European pilot line dedicated for photonics devices. And we are going to work on this project for 4 years. It started beginning of this year. And the end the end goal here is to really have a sustainable ecosystem and pilot line environment that startups, companies, research institutes can go to accelerate the commercialization of their devices. So the main benefits of working with us, I think that's maybe an important angle to talk about, is that typically when you're a small company or a research institute, you have to find your technologies puzzle piece by puzzle piece. Very often you're standing there, you're dealing with a lot of partners, a lot of potential opportunities, but in the end, you're gonna take your individual technologies, upscaling options and so forth together like a puzzle. And that can be very consuming and time intensive and thus expensive. The idea of MEDPHAB is that we pre-arrange these puzzle pieces for you. So we work with currently 18 partners in a centralized community, partners that already have a very strong track record, and most importantly, partners that already know what the other partners can do and what they cannot do. I think the strength really of this pilot line is that we are establishing at the moment a so-called technology map or innovation maturity map, where we exactly map down the individual competences of all the partners. We will rate them in TRL levels, and something new that you might have not heard of is MRL, manufacturing readiness level. So when companies are coming to us and they want to have a certain technology problem solved, we can direct them very fast to the correct partner and speed up the commercialization of the project. Here, a quick peek on the current partners that are part of the project. So in general, when you talk to us, to MEDPHAB at info@medphab.eu, you get all these brains thinking for you for your project. And I think that's a very, very unique offering. And I'm going to tell you in the following slides on how we will realize this in the upcoming calls. Okay, shortly about the different technologies that we are offering within the pilot line. This is a small selection of some of the main technologies that we have as an offering from our partners. I think the important part here is that we can cover the whole spectrum really from R&D services in early development all the way to ISO 13485 certified manufacturing. I think this is very often a gap where technology is getting stuck. That's, for example, if you're working predominantly with RTOs, for example, they can only go so and so far. If you work with manufacturing companies, they might not have the manpower and willingness to actually invest a lot into early stage R&D. At MEDPHAB, we can cover basically the bigger picture. So how to work with us? So the idea is that, and this is, by the way, all in establishment at the moment. We are just in month 11 of the project at the moment. So we don't have a full functional pilot line yet. I think that's important to mention, but we are very interested in talking to you already. And you have, through your input and through your discussions that we can have with you, a good chance to also shape how the pilot line will look like. The general idea is that we have customers, any size company basically works, who contacts then MEDPHAB through a centralized front office. So there will be one individual contact point, and we will go with you through your technology brief. So we will generate an intake form or an intake document where we specify the high-level goals, what you want to achieve. And then with that intake form, we are planning and mapping the technologies that we have inside MEDPHAB. So we look at the individual partners, what their competences are on a TRL level and on a MRL level, manufacturing readiness level, and then we get back to you in relatively short time on the technologies that we can help you with, but also we try to identify then technology gaps and where we need external partners. And of course, we also collaborate a lot with the other European pilot lines. So shortly about the positioning of MEDPHAB. So this is one of the graphs I think that we have found very, very useful in early discussion with companies. So basically, as you go along, I hope you see my mouse cursor here, as you go along the technology development of a project, you get more expensive in project volume. The more you're going into a controlled DMR environment, the more you're going to pre-manufacturing, manufacturing, clinical trials, the more expensive development typically gets. Here in the beginning, you are still very flexible, but to be able to transition really from early stage development to manufacturing and productization, you need to be able to follow certain quality and regulatory principles. And that's what we can help you with. So we have companies such as, for example, Philips Innovation Services and Jabil on board that do manufacturing all the time. And we can help you to create the experimental part and the upscaling of your devices in such a way that we can blend into regulatory then towards the end of the project. As you can see, MEDPHAB is covering the middle ground here. So we are not taking care of final manufacturing. This is not the case, but we are identifying manufacturing partners throughout the project who can then take care of the final manufacturing for you. I want to quickly stop on this slide here, because I think this is maybe the second most important slide that we've been using now in early discussions with customers. So when you come to us with a technology need, and we write down and discuss with you to get the question right, basically, what is it what you need, what you expect from MEDPHAB. It's very important to put down the project roughly on the map, to say, for example, we are now here still in a very exploratory phase, and we try to get within the MEDPHAB project, or with help of MEDPHAB, to an advanced development stage, for example. Or we are already very advanced developed, and we are aiming at product launch. So mapping the project on this slide, I think is very, very important in the beginning, to also know what to expect throughout the project. MRL level, I think, is something that very often is neglected. And I think this is one of the strengths of MEDPHAB, in a sense that very often you might have components or devices that you've been working on for many, many years, which have a very, very high TRL level already. And I think it's very often companies think or believe or hope that if you have components that are very high TRL level, that basically integration of making a product out of it is very easy. But the reality is that the integration level needs to be developed as the TRL level as well. So advancing technology on a manufacturing readiness level scale is something we can help you with at MEDPHAB. And we can give you a realistic estimate on where your project is currently, or where your technology is currently positioned within this graph, and how far we can get on an integration scale as well. I want to show you a short example. This is actually something close to my heart here. So we have been developing here at VTT a fluorescence reader, a miniaturized fluorescence reader for lab-on-chip devices. Basically, it's a wireless reader device that has very sophisticated optics inside, and it sends then the data to the mobile phone. And it can read microfluidic chips or lateral flow tests alike. The issue with this is that it looks like a product, it behaves like a product, but it is not a product. So we got at VTT only so far to basically develop everything around the disposable chip. We have developed the optics, the electronics, the wireless communication, the app, and so forth. But we cannot work with real clinical samples. So we've done some preclinical testing now with biobank samples here from local supplier. But to get to ISO 13485 certification and to production, we would have to advance this device on an integration level, maybe from MRL level like 5 to 9, for example. And this is something that MEDPHAB, for example, can help with. So here are some extra details on the readout principle. That's maybe not so relevant here now. But if this case would now come into MEDPHAB, we would now map all the technologies on where they are currently. And the goal of this device could then, for example, be we want to go from preclinical testing to clinical testing, and what partners could help us then with upscaling of the device, production of the device, and certification of the device. This is now a slide that you probably should take a screenshot of or note down. We have built in our pilot line project 2 open calls in June next year and in June in 2022. We will have open calls for projects from external projects that we will pilot inside the MEDPHAB pilot line. So we're going to be in year 2 and year 3 of our project. And these cases are meant to be there for testing the front office operations, the customer intake, and the whole process really from working with external customers. Bear in mind, so we want to have a fully sustainable pilot line at the end of these 4 years. So it's a pilot line that should exist way beyond the lifetime of the project. And these are the 2 open calls where we'll test the operational part. If you're interested in participating to these calls, by the way, there's a cost sharing model implemented here. So the European Union will carry some of the costs for the external customers. It's not quite clear at which percentage. At the moment, we think somewhere between 50 and 70%. But if you want to stay informed, check out the webpage www.medphab.eu or send us an email at info@medphab.eu. That's where I'm also then can get in touch with you. So I'm seeing your emails when you send it to this email address. And yeah, we're looking forward to collaborate with you. So I think my time is up, and I'm looking very much forward to the collaboration with you.
Thank you very much, Ralph. Thanks a lot. So you were exactly on time, even a couple of minutes earlier. So we compensate for my too much talking at the beginning. So Ralph, it's really interesting. You know, I have still my heart in MEDPHAB because I studied the project before Lena, my colleague, was studying took over. So for me, it's really interesting to see this actually manufacturing readiness level is really a nice point for MEDPHAB, because maybe you can spend a couple of words more there. I mean, you put already a lot of attention there, but basically if a project comes and you will evaluate also this readiness level in this direction, and then it will be taken over by a specific partner, or how will it be then really driven?
Yes, so the process will go that we will have a customer intake form. At the moment, it's still a Word document. In the end, we will hopefully have a web page where external people can contact us. And then we go with a dedicated expert group through that individual proposal or through this technology request, actually. And we will have in the background then our technologies of all the partners mapped out with all the TRL and MRL levels of all the processes. And then we try to put that project on the map. We can identify then rather quickly. These would be, for example, the 3-4 partners that would be ideal for you. We also will have so-called production kits. So some of the technologies that are going together very often, for example, fiber optics integration, PCB design, and so forth. Those will be production kits of pre-arranged partners that have been used before. So basically, in the analogy of a puzzle, these are pre-assembled puzzle pieces that we can present to you. So you don't have to take care of the whole picture anymore. And then at the moment, it's not 100% decided yet, but the way of then collaborating would be then you will have a prime collaboration partner. So you will then contract with one company, and the other little bit lower priority partners will be then working as a subcontractor. So MEDPHAB is not a legal entity, it's a project. So as MEDPHAB, we can not make offers. We are not a legal entity, we are not a company, but the partnering will be then through a prime partner from the MEDPHAB community.
And do you plan to make a legal entity at some point, maybe after the duration of the project?
That's to be decided.
Okay, so still open. Yes. So very good. So thanks a lot, Ralph. So hopefully everybody took a screenshot of that of the dates. So we are really soon open for calls, and so hopefully we will have many applicants. Is there actually, Ralph, some rules about applications? So there is someone that is excluded or recommended to apply, just to tell to our audience?
Not sure what you mean with this, but if there's, for example, now a company and says that, wait a second, one of these MEDPHAB partners are our direct competitor. We do not want to share any information with them on our technology need, then this is possible. So you can, for example, exclude the sharing of this intake technology request form during filling out the intake. So this is
okay, but from your side, nobody's excluded, so everybody can apply with this project or her project, and then come. Yes, so this, okay, very good. So thanks a lot, Ralph. So now, just to give you the overview now of the agenda. Now it's time for Professor Dr. Fokko Pieter Wieringa. So welcome also to you today from imec, Inter-University Microelectronics Center from the Netherlands. Today, he will talk about medical photonic technologies to embrace a better life. Thanks a lot.
Okay, I hope I'm hearable and visible at the moment.
Yes, hearable, visible not yet.
I have stopped and restarted my video now, so now everything is stopped. Can you still hear me?
Yeah, now, yes, we also see you. Now,
okay, well,
very good. You can share your screen.
I'm going to share my screen and let's see what happens. Can we see the presentation? I hope the presentation is visible.
Yes, it's there. The floor is yours.
Good. Then let's get going. Okay, today I'd like to talk to you a little bit about what's going on here. Is the screen...
Now it's back. Now it's back. No worries.
Today I'd like to talk a little bit about how we get things, photonic chips systems, from the clean room towards the bedside, and how imec might help you on that. So first of all, what's imec? Well, we're one of the proud partners in MEDPHAB, as you saw. So we're really happy that we're in there. Our headquarters is in Leuven in Belgium. And we have a lot of people working in clean rooms, making chips and designing those, and looking upon how we can make key enabling technologies, how we can create those with chip technology as the core. Okay, that's one thing. Now, I work very close to Leuven, but in the Netherlands, in Eindhoven, and I work on applying key enabling technologies in medicine. So I try to take the great new stuff that is created in our headquarters and try to apply this in practice. So one of the examples of things that imec does on photonics range, for instance, is expanding the spectral range that can be achieved with CMOS technology without leaving the fab. So in one fab, different processes combined, CMOS, and then, well, you know, it starts for me when the engine seems to be running and I have to go into the clinic and realize practical applications. So from clean room to clinic and as fast as we can. So one of the examples today is how to replace and quantify the qualitative fingertip feeling, Fingerspitzengefühl, as the Germans say, that the clinicians have when they're feeling your pulse. How can we quantify that by medical photonics? And I must admit that this is not my idea. It's from Professor Roel Baats, my colleague at imec, who's also working at Ghent University. He had the idea to take the Star Trek tricorder, the contactless scanning of Dr. McCoy, and put it into a kind of a pistol with photonics. And he actually did it. He wants to use, and he is using pulse wave velocity as a marker for arterial stiffness. Well, as you see here, this is an artery that is healthy and then is progressively getting more stiffer and sicker, and cardiovascular disease is really serious. It's a big killer. About 1/3 of the killing by disease in the world is by cardiovascular disease. So it's a serious thing. And pulse wave velocity, as I say, is an indicator of arterial stiffness and the risk on cardiovascular events. So how does it work? Well, the heart beats, pumps the blood through the body, and that gives you a pressure pulse that is traveling with a certain speed across the arteries. And this traveling speed says something about your health. So how does this look like in practice? Well, McCoy would have liked this. This is an optical gun that is projecting two beams with a known position, a known distance between them at the carotid artery that you can feel if you're a bit experienced and you do first aid, you can feel that. And with laser Doppler vibrometry, you feel the pulse without touching it because the light is feeling the pulse. How does that look like? Well, here you see two signals, but I must say they're very close together because we only have a short distance. So if we zoom in, we see there's a time difference between those two signals. That time difference tells you something about the speed because we know the distance. How does it work? How is it built? Well, we have 6 laser beams coming out of a photonic integrated circuit, a so-called PIC, with a lot of components, optical components integrated. Under the microscope, it looks like this. The laser is input here, and you get the in and output. So this is a send and transmit antenna optically, and it's all on a 2.5 by 5 millimeter chip. That's quite small, but to make the gun, the chip has to be embedded into a housing and has to be on a micro-optical bench, and that's all depicted here. So you get the vibrating target, which is the skin, and then you see this. Well, this is how it looks like from the side, but the fun thing is you can split this gun in 2 halves, and then you can use it not only with a known small distance on the carotid, but you can also use it on a longer distance between 2 anatomically very well known, by doctors very well known places where you can feel the pulse. So you can choose where do you want to measure, as the existing devices normally do as well. So as an example, here we have a bigger distance, so also a bigger time difference between the pulse wave travel. The bottom here is ECG, that's the electrical heart function, and here we see the pulse waves arrive at proximal and distal. And from that, you can calculate the pulse wave velocity. Now, we compare here this Cardias gun, handgun, optical gun, with a commercial sphygmocore pulse wave velocity meter. And this is quite a very good correlation. And now we also looked upon, if we take a commercial bedside monitor that needs ECG, so with stickers on your chest to calculate pulse wave velocity and compare it with our principle that does not need ECG, how does it look like? And this is the line that you see then. So without bothering a patient by sticking stickers for an ECG, you can measure pulse wave velocity quite well. That's interesting. Now, totally different topic, because I promised you several topics. This is a chest patch, as you see on the chest of this pilot. And the pilot takes off, and we record with a finger probe, which is a bit cumbersome for the pilot because, well, you use your fingers normally during work, but we compare a commercial finger probe with a chest SpO2, and what we see is that it's creepily good. No matter how the guy rolls and rocks and rolls with the plane in the air, it goes quite well. This is a lady that has a more steady life and is more calmly resting, and she also has this chest patch. It's very small, and this is electronics all on a very small chip, and the photonics are here connected to the PPG signal, so a photodiode and a LED, and it's all integrated. You have a complete bedside monitor, but it's all in a sticker. Now, totally different example again, measuring the sweetness of life. How do we do that? Well, imec is a at the bottom, at the foundation, together with Ghent University, of Indigo, that made a continuous glucose monitoring with a subcutaneous implant. You see the implant here, and it contains an implantable spectrometer. So it's an optical spectrometer that runs for at least 2 years, 2+ years, while being very slightly under the skin implanted. It can communicate with a readout simple sensor here that you can clip on any place that you can see it, how do you say it, but also with your phone, and it records the trends. This is a joint spin-off from imec and Ghent University. And this is how it looks in practice. You see the readout unit here in the pocket, mobile phone. These guys have an ISO 13485, as we just heard from MEDPHAB, very important. You can contact them if you want. I have no shares in the guys, but I do think it's a quite interesting principle. It's co-funded by imec Expand, and it has been tested in pigs already, and it looks extremely well so far for that example. Now, another totally different example, photonics in the operating theater. This is a stereoscopic head-mounted microscope with a full electronic chain. We know optical microscopes as a lens system, but this is electronic. So it's a camera plus near-eye display all in one, and a small computer. Now, what can we do with that? Well, as we know, the naked human eye can only see the visible range. That's the definition of the visible range. But we know that a camera chip can have a much wider range. So that is the trick. You add a wider spectral range to the eye of the surgeon that then can see things that are invisible to the naked eye and make surgical guided decisions on that. It's an attract program that is running at the moment, and we're looking for partners for the second round. It uses a small belt-worn computer, which is customly designed for this. It has a wireless foot switch. It has 3D monitoring also, so the assistant can look through the eyes of the surgeon with full 3D instead of looking across the shoulder. And with the COVID thing now going on, it's not only for teleconsult, but also to keep the education of surgeons in training, medical students, to keep their training going. So they can now look virtually through the eyes of the surgeon that does the surgery and explains why and how she does it, while the students really look through her eyes. And even it could be in a classroom, but due to COVID, that will follow later. But it's already being used in the Netherlands now to keep the anatomy and surgical education of medical students going. Because normally they would be crowded in the OR, trying to look across the shoulder of the surgeon to get a glimpse of what they're seeing. Now they can see through the eyes, and they can even record it. I'm very enthusiastic, sorry guys. Spectrometry. Okay, now what can you think about PPG? And we all know this finger clip for the oxygen on the finger in the hospital, but what if you would have a full spectrum of that? Well, that is a dream that I had for decades, and it's becoming a reality. Spectricity is making this. You get a fast spectral sensor at your fingertip. This is the sensor, and it's a true spectrometer, and it is fast enough to get the pulse wave. So you can imagine what you can do with that. You can do a lot of things like heart rate variability. You can look for spectral components in the fluid, the water content. How's my perfusion? The respiration rate can be easily got from it. Blood pressure could be done with the pulse wave velocity, but I will focus on SpO2, the oxygenation of the blood. How much oxygen is in the blood? They already have a wrist-worn evaluation module, and they tested this very nicely according to the ISO 80601-2-61, as everybody knows, of course. And compared to the commercial equipment, it looks very good. In fact, compared to other smartwatches, they have a 1.5% less error margin. So they have a more narrow margin of the signal, which is, I think, a very good prestige. So I hope these guys will get a lot of exposure, and this component will appear in many, many wearables. Now, to make this more or less short, by the way, if anybody wants to contact anybody, the name of the person, the expert that I'm talking about is always in the left bottom of the screen. I would say to everybody, you know, keep your eyes on medical photonics because it's going to be booming. What electronics have been doing for mankind for the last 100 years, well, photonics, at least for the coming 50 years, will do for mankind as well. So keep your eye on photonics, but keep your pupils vexed. This is an imec electronic pupil contact lens. So with LCD, you can vary the diameter of the pupil. This is an outcome for people that have a non-functioning pupil, but still a retina that works. And so they can then get out of the dark, literally, and they can get rid of their shaded glasses and they can live in normal life. That's also another example of photonics. And with that, I think I can say, well, normally I would say thanks and bye, I have to fly, but this time I'm seated in my music studio at home and open to any questions of anybody of you. Thank you very much.
Thank you very much, Fokko. This was a great overview of what imec can do. So it's really great that you could give us this. Yeah, we flew at least virtually on the topic that imec is treating. So as I said, we are taking questions after every contribution. So I have already a question, so I have to for sure to say it. So this microspectrometer is really actually really cool. I mean, you really show really nice technology, but this one is for me a bit striking because we have for a complete different application another European project where we use microspectrometers for optimization of processes, of industrial processes. So these are really various applications. But for this now that you showed, we see also in general the readiness to the market of the products that imec in general produces as on this spectricity. We saw that some have a spin-off connected to them and some are still imec core. So is this connected to also the readiness of the product to market, or what is the procedure that imec does with the product?
No, indeed, you're totally right there. So imec encourages always that technologies are reaching the market, but we as imec ourselves don't make any products. So that is the reason that, for instance, a firm like Spectricity with that small chip on the finger, which is a complete spectrometer, that is encouraged to go to the market. They are a firm. They are having this at not only a technology readiness level, which is high, but also indeed a manufacturing readiness level. It's a good one that we shall hear again from MEDPHAB. So I like that very much indeed. And that's the idea that spin-offs are encouraged from imec, and we see spin-offs growing, and sometimes that's the biggest reward. Spin-off companies grow so much that they become our customer later, and that's fun.
Yeah, that's really great to know. And also it means that what you bring out can also be used in other technologies. So for example, your suggestion on spectricity is for this measuring oxygen level and saturation levels that actually is good for corona diseases, right? This was not by chance. No. A very good example, because it's something that is really important to measure, right? When people get to this level low, then they should be brought to the hospital. So that's really nice, also wearable. So thanks a lot, Fokko. So it was really a nice overview. Thanks a lot. So don't leave, stay here for future questions. Who knows, people can ask a bit later, maybe other questions to you and to Ralph as well. So now let's go on with the agenda that I this time will share for real, and I just won't tell you only because I guess before I didn't show it to you actually. So there it is again. So we are now at the third contribution. So it's really a pleasure to present you Dr. Dominic Zerulla now from PEARlabs in Dublin, and his talk is called Making the Invisible Visible, very attractive already, and a new real-time imaging method beyond the diffraction limit for the life sciences. So Dominic, please, I leave the floor to you now. Stop sharing, and the floor is yours.
Thank you very much, Francesca. Yeah, thank you very much for the two talks. I think they were really nice and really cool. I mean, I like Ralph's manufacturing readiness level as well, because I might connect to that at a later stage, because actually this is exactly what we're facing here in our development. Okay, let me screen share here, and. Okay, so that is the presentation. So what I'm going to talk about today is about a new imaging technology. So what we have developed is a new, radically new, completely different imaging technology, which can provide imaging beyond the diffraction limit of light. So I think we all know what the diffraction of light is all about, because they're all photonics experts here, but I'm going to, of course, to introduce the technology itself, maybe in 2 or 3, 4 minutes. The idea which we have behind that with this new technology, we have a couple of key advantages which the existing super resolution techniques don't have. Okay, let me jump into the talk. Okay, so this is the only imaging method worldwide which is capable of imaging nanoscale structures at video rates. I mean, yeah, I know there are a couple of techniques which you can scale up and you can try to make them very fast, but most of the super resolution techniques are actually either scanning techniques like STAT, for example, or they are actually techniques like PALM or STORM or variants of this, where you have to make multi-acquisitions, so thousands, 10,000s of acquisitions to get a very good image. And that means that in the end, either you throw away most of the photons in one particular picture, or if you're scanning, you have basically a single channel technology, and that means the throughput of your technology is quite low. So therefore, you're usually limited by signal to noise ratio problems at some point, or you have to blast the sample with such a high amount of photons that basically destroy the sample, which is not good for a real-time video imaging method in the end. Okay, so so we have invented a entirely new way of imaging in that sense, and PEAR is actually an acronym. So we're not talking about the fruit here, but what it means is actually it's a plasmonic electronically addressable super resolution technique, and basically in the acronym you see already a little bit of the principle behind it. And this is, we have electronically addressable pixels, and these pixels are not normal pixels because they have to operate in the near field regime. So actually what we have are little small plasmonic pixels which are basically electronically addressable. And that means that we can smuggle the information from the far field, or from the near field into the far field. Okay, so basically this idea is based now on a couple of almost 25 years of experience in the field of nano-optics and photonics, including some work I did myself at UC Berkeley in 2013. And so the company PEARlabs itself has been founded in 2018 actually to commercialize just this. Okay, so just to bring a little bit of a perspective to the whole thing. So as we know, we can see all the things with a normal microscope to the left side of the dotted line here. So we can see cells, we can see parts of the cells, but what we definitely can't see are details of the structures within cells, which are part of the organelles, for example. So we can't see viruses, which is quite a problem at the moment. At least we can't identify them with optical microscopy in an easy, direct fashion. And we surely can't see smaller structures like protein structures or even small molecules, which is quite, in fairness, a challenge. But basically, the Abbe diffraction limited is roughly at 200 nanometers. Yeah, some methods like confocal microscopy can be a little bit lower, so they can provide maybe spatial resolutions around 150 nanometers, but that's basically where it ends and where the super resolution techniques actually start. Okay, and obviously, I mean, it's not just because Corona, but in order to understand even cancerogenous processes, so how cancer proliferates, how cells actually, how are cells reacting, for example, to small particles, and all these questions, which are very important for biomedically relevant samples and diseases, you cannot really investigate them directly. Yeah, we have electron microscopes, for example. Electron microscopes can image down to sub-nanometer resolutions nowadays. But the point is then you have the problem that you don't know really, is my sample still intact? Is the sample still working? Is it still alive? And normally, if you have to use vacuum, even in these ESAM conditions, you have killed your sample, or at least you have strongly modified your sample. Okay, just as a principle, this is what STAT is doing. STAT is basically having here this, yeah, in the end you have this orange donut mode. Sorry, actually, this is not how it should work. Go back. So I just wanted actually to highlight one or two things here. So basically what we see here is this is this orange donut mode, and this orange donut mode is depleting an area surrounding the area you actually want to see. So you basically have one laser pulse which is depleting the area around that, and then you can interrogate the smaller area within it, which is the blue laser beam here, or shown here on this side here. So you have an excitation laser beam which shows what has not been depleted, and the trick here is that the depletion zone can be smaller than the fraction limit of light. However, what you have to do is, of course, like a little bit like a potato stamp, you have to stamp your picture with your laser, with the two laser beams, or the two laser pulses, I should say. And that is very small and has an inferior signal to noise ratio, because it's basically just a very slow technique in comparison. Okay, so what we are doing is quite different. If you first look here on the left-hand side, what we have created is an optical chip, and this optical chip can actually, you can address the optical pixels on that. So what you can do, for example, as shown here, you can switch on light on a sub-diffraction limited scale. So for example, here we have used pixels which have a 40 by 40 nanometer size. And these pixels then, if illuminated from the backside under plasmonic conditions, and when the conditions are met, in the end, due to the electronic addressability, then actually what you see is you can create a very sharp, confined illumination of your sample. And obviously you can control this illumination. So actually what I show here is actually a series in time where addressing has just started, and then we shift the conditions more and more into the resonance conditions until we get a very, very sharp peak, which is subdiffraction limited itself because it's a near field process. The real advantage is now, as you see on the right-hand side. So what we can do is we can switch on all the pixels. And this is just a simulation. So obviously we have more pixels than just the 25, I think we have here. So we can have a megapixel chip, for example. And this megapixel chip allows us to switch on and off all the different pixels at will. First, that doesn't sound maybe like a multi-channel technique, but the trick is now that we can modulate them with a particular frequency. And with a lock-in heterodyne detection scheme, we can automatically filter out which pixel, so which information was illuminated by which pixel. And that can be mapped into a super resolution image in the end. Okay, so we have a couple of use cases for this. So basically, one is the obvious one as a researcher. We can provide a new tool for state-of-the-art research, and we are already working with a couple of clinics throughout Europe, actually to demonstrate what our new technology can do. So we have reached spatial resolutions. Actually, we are aiming for below 20. We have reached 21, as I can show you on the next slide. But actually, more importantly, we can provide real-time acquisition. So we can provide real-time acquisition at the same time. And what we're providing, among other things, is we have that in a biocompatible environment. And the function actually doesn't come from a huge microscope, because the real spatial resolution comes from the little optical chip. And that means, apart from a quite bulky large microscope, you can also think about actually putting a rack and put hundreds of these chips into it, which actually creates a kind of a multi-microscope here with an automatic feeder system, and artificial intelligence can actually analyze samples. And that can be done so quickly that this is actually really accelerating drug testing and drug delivery. Or alternatively, you can actually expand this idea and actually to make it directly a mobile device. So you can have a little platform, a USB connected platform, which can be connected to whatever, to a tablet or to even a phone. And you can actually use the chip as your personalized medical diagnostic device. And it can be used for remote screening, for example, in places where a doctor is to remote and you actually you want to send certain images of your body or certain images of your blood or of saliva or whatever to the doctor for fast screening, because you might feel that there's something wrong. So basically, if you like, this invention brings us three use cases. One is the classical state-of-the-art research, drug delivery is a second one, and then a mobile device is the third one. Okay, and this is one of the results we're very much proud about. What you see here on the left-hand side, this is a picture of nuclear pores. So they are core structures in cells. And you see this is actually done with a high numerical aperture microscope. And basically you see not much. So this is these structures, you see this is just across, this is 200 nanometers. You see a little bit of the structure here if you're generous, but you don't see any details. If you do the same sample with our super resolution imaging, and we can switch between the two, because here this is integrated into the normal microscopes. You can switch between normal microscope mode, if you like, and between our super resolution. And you can clearly see now, you see even details in the core structures, which are confirmed by other technologies. So what we can do, we can at the moment resolve roughly at the 21 nanometer spatial resolution tag, but we do that at the moment with 10 frames per second. And if Corona wouldn't have been in the way in delaying us, we probably would be already at 25 frames per second at that stage. There are 1 or 2 items which haven't been delivered in time due to the Corona crisis, but we're quite positive that this will happen soon. Okay, so if you talk about the market for this development, actually, we talk about the market, the whole microscope market is around $8 billion for 24. And the super resolution market is a growing part of that, which is around 1.5 billion for 24 with a capital growth rate actually of 11%. So this is quite nice to go in such a market. This is the team. So we have a team of young researchers in the middle bar, and then we have a team of more experienced, actually, experts for commercialization, including 2 people on the oversight board, Dr. Conor Henley and John Brown, who are CEOs of established companies. And let me just maybe reiterate some of the advantages of the technology. First, it's compatible. So basically, you can even retrofit that into existing microscopes. That's quite important for our starting point, because you can go with minimal investment, basically. We have very high spatial resolution, so this is a given one. Sub 25 nanometers have already been reached. And this is actually something I should actually now bend back to the first talk of today. You know, an institution or let's say a research project at the moment like MEDPHAB is really needed, because as the first speaker Ralph has already said, there's a huge difference between the TRL level and the MRL level, because the ability actually to manufacture them is different than actually making the prototypes. So we can make prototypes, but we can't mass manufacture them at the moment, at least not in an easy fashion. So this is what we're working on at the moment. And of course we would appreciate any help we can get in that. So this is a very fast technology, because it's basically multi-channel up to 1 million channels in parallel. This is a fast video array technique. It works, it can work label free. Well, the picture I've showed you actually used labels, because we are free to use labels, but strictly speaking, we don't have to use labels. And it's biocompatible, because it's a complete normal optical technology. It doesn't require vacuum, doesn't need any cryo freezing. It can actually be done on special Petri dishes in which the cells can even be grown directly on top of that. And it's relatively affordable compared to other technologies. Okay, so maybe that's the point. I mean, I can just proudly announce that we got the highest national award in Ireland, though the Future Innovator Prize, which has been given to us in May this year. So we are quite proud of this. And maybe that shows that we really have a very nice, but also important disruptive new invention, which should be carried out into the photonics world globally. Thank you very much.
Thanks a lot, Dominic. It's really great what you presented today. So it's really impressive what you can do at the PEARlabs. And so all this possibility also to expand the technology. So in different, the 3 options that you gave us. So my question is more related to what you said at the beginning about the throughput. So this is of course fundamental, that is high if you can have really an online monitoring going on. So is it actually the image processing something that you worked on, so to make it faster, or is just that the images are so optimized that the size is easy to be handled?
Okay, I'm not sure I fully get the question. I mean, I understand the question, but in what sense? So I mean,
if it's more on the purely, let's say, IT handling of the data, or that it means that you have a specific code that can make this image processing fast, or if it's also the image size and all the optimization that you implemented in the technology.
Well, first, I mean, it is quite unique in the sense that it can actually measure all the information from all pixels are measured in parallel. So this is like a CCD camera almost, okay, in that sense. It's just not a CCD camera, it's quite a different thing. So the way the pixels are designed and the way the pixels are basically addressed are quite different. And basically what we're doing is we are modulating the sample. And then you're right, there's of course IT needed. So there's quite a lot of data we generate. But the bottleneck is actually really the signal to noise ratio. So if you make smaller and smaller pixels, they illuminate a smaller and smaller section. And if you make them smaller than 20 nanometers, you find, of course, a place where you might only get a few molecules which are basically, for example, which fluoresce. And if there are just a few molecules which fluoresce and you want to get the modulation of that, then of course you run into a signal to noise problem in the end. But at the moment, you're right. What we're doing in order to come from 10 frames per second to 25, this is more actually really a computer and computation issue. But the fundamental limit is actually really signal to noise, capturing enough photons from each of these very small sections to be processed in real time. And this is also the limit on faster frames per second. So if you want to, in theory, you could do 10,000 frames per second, but then if you do that from a very small portion of the chip, then you don't have enough photons probably to do that. So there is a limit there. And we, of course, would like to see where the limit really is. So that's not clear.
It's open then for you to find it, this limit. So, and then maybe you will need help, and then you can ask EPIC to help you to overcome this limit. Don't forget. So now it's time for Fokko. There's a question. So now if you unmuted yourself, Fokko,
yes,
you can ask the question yourself. Yeah.
Okay, Dominic. So this is extremely exciting, very, very nice work. And it by coincidence, matches extremely well with an ongoing consortium that we have on regenerative medicine in Europe. And I would think that this might be a totally good add-on to watch our cell cultures growing while we are, working with the stem cells to make them grow into the organs that we'd like. So would you be open to consider talking with us about that?
Oh, I would be delighted to talk about that, because I mean we're looking for investment from large companies at the moment, and of course they also like to see that we have as many use cases as possible. And actually, I'm as a researcher myself, I'm quite intrigued to apply our method to interesting systems. And what you said is actually sounds very interesting.
Yeah, well, thanks. And I must also say, as a guy that has been working on the optical radiation levels, the intensities that living tissues and cells can bear with ICNIRP, the International Committee on Non-Ionizing Radiation Protection. So I know that you cannot put an infinite limit of photons on cells. They will be killed simply by the heat. So that would be a very good one to see how we can maximize the yield of each photon that goes into the cell without killing the cell or altering it. Thanks very much. This is really exciting stuff.
Thank you very much. I mean, I really appreciate that comment.
Great, Dominic. Then also, I remember you mentioned that the MEDPHAB could help you as well. Can you repeat maybe this point? Because this now I forgot it, and now I have to tell my colleague when I'm done. Okay, there is also Ralph still online, but let's repeat how MEDPHAB can help you.
Yeah, I mean, look, we're making this optical chip, and what I found out, which was of course clear to some extent, but if you go to a very small nanometer scale, then you find out that most people don't handle optical components. So, I mean, materials which are optically useful. So all the people come back to silicon, and we all know that silicon, I mean, we have just seen that TSMC is outputting chips now with a 5 nanometer node, which is quite a strong message, but they are only used to really work with electronic systems. So whenever you switch over to say, okay, can we switch this material against a photonic, and some material, even like a transparent material as a top layer, they already run into problems because they have never tested their E-beam lithography and all the other techniques they're using to make the small structures. So I hope that MEDPHAB could be a super hub in order to help us with exactly these problems.
Yeah, Ralph.
That would be great. That would be great. I have to say, yes, use the intake form. So we would love to go through your case as well. So let's be in touch. If you send an email to info@medphab.eu, I will see it. Yep. And maybe we can use your case also as a kind of a guinea pig case to actually make our pilot line better.
Yeah, that would be great. I mean, yeah, I've spoken actually to some person in Finland. I think Matti, but then the family name doesn't come to me, so you probably know him very well. And we had a kind of an initial talk about that, but I like to go the official route. So I'll send you an email, Ralph.
Perfect. Thanks, Dominic.
That's very good. That's what we want to hear. You know, we are still, it's still an epic TechWatch. We want to, we want to see that people collaborate. So really nice that that this can also be done with our pilot line MEDPHAB. So very good. So now it's time probably to go on. Thanks a lot, Dominic. Again, I guess it was really fruitful showcase for you, and we get also more opportunity of collaborations also with Fokko, and who knows who will contact you after this. Thanks a lot. So now we go back very briefly to the agenda. There it is. And yeah, there it goes. So we are at the end of the first page. So it's now time for Professor Dr. Werner Mäntele from DiaMonTech in Berlin, in Germany. So his talk is called Non-Invasive Glucose Monitoring for Diabetics based on IR quantum cascade lasers and photothermal detections. Thanks a lot, Werner, for being here. And now the floor is yours.
Okay, can you hear me?
Yes, very well,
okay.
And we can see you.
Let me share the screen.
Please.
Okay, can you see my screen?
Not yet. There is this button on the green button on the bottom.
Okay, okay.
That is probably for you also Bildschirm freigeben.
Yes, but let me see, here we are. Okay.
Yes, now it's coming slowly.
Okay, now let me go to the presentation mode.
Very good.
Okay, presentation mode is on.
Yes,
yep.
But maybe, do you have another? Now, yes, there you go.
Okay, very good. Let me go to the start. I'm pretty much at the end of the presentation here. Hi, everyone. My name is Werner Mäntele. I'm the CSO of DiaMonTech. DiaMonTech is a company that we founded in 2015 with the goal to develop a non-invasive glucose monitoring for diabetes patients. And DiaMonTech actually continues the R&D work that I did for more than 20 years at Frankfurt University, where I became emeritus in 2018, and now I'm working for the company. The goal is really to create a real non-invasive technology for glucose monitoring for diabetes patients. And let me just briefly introduce you to the topic. Diabetes is really an enormous problem. If you look up the standard reports of the International Diabetes Foundation that come up annually, then we had about 460 million people in 2019 that lived with diabetes. And unfortunately, this number is increasing and increasing. So it's going to about 500 million in the next 20 to 30 years. And it's globally a bit different, but the Western countries are really at a very high rate of diabetes. Actually, also pre-diabetes, people that are not yet diagnosed as diabetes patients, but these people will become diabetics in very short time. Now, you all know that diabetes cannot be cured actually, but the management of diabetes is possible. And the standard procedure of managing is that measuring your blood glucose level several times a day, and in dependence on the glucose value of your blood, either injecting insulin or changing your food intake or your physical exercise. These test strips have been around for more than 30 years. They work nicely, but they still require a drop of blood. They have a limited shelf time, and their precision is not high enough really to adjust the blood glucose value. For the past 10 years, there have been continuously measuring devices that are actually little patches with a small needle that can stay for up to 2 weeks in your muscle, and that allow a more or less continuous measurement, but these things are not very precise, and they have the problems that lots of people have allergies against that. And all that amounts to an enormous amount of costs for our health system. Just for example, in Germany, we have about 6 to 8 million diabetes patients that measure about 10 to 15 million times a day. And those costs are enormous. And if you do a Google search, diabetes worldwide, you immediately find these headlines that diabetes is a global problem, a challenge for, and a worldwide national threat. We shouldn't forget that in the present Corona situation. So our goal is to develop a reagent-free and non-invasive glucose measurement system for diabetes patients on the basis of infrared spectroscopy. Now, why infrared spectroscopy? This is a long research from my group and from other groups that shows that almost all molecules in our body fluids have clear infrared signatures due to their individual bonds and vibrations. And these infrared signatures are, just an example here, are very pronounced for glucose. They are located for glucose in the region around 10 micrometers, which is about here. So deep in the mid-infrared, and all these peaks are actually related to different CO bond vibrations and OH bond vibrations that couple in a very specific way to form a fingerprint. And it is this fingerprint that can allow a very selective analysis of the glucose level in body fluids. Now, if we take that as a basis, we can ask the questions, what body fluids are actually representative for blood glucose? Well, blood is there. Whether you take venous, arterial, capillary blood, you find only very minor differences that, so each of these blood samples can be taken. Mostly people take capillary blood from a drop of blood from the fingertip. That's the standard way of doing it. Urine is not suitable because it contains glucose only for a very high blood glucose level. Saliva turned out to have traces of glucose, but without a fixed correlation with blood glucose. Same for sweat. And tears have been analyzed a lot. They contain small amounts of glucose, but they don't have a fixed correlation. And even worse, the glucose in tears shows up with a strong delay after blood glucose. So if your blood glucose varies, your tear glucose could be varying like half an hour, 1 hour later, which is a problem specifically for type 1 diabetes patients. Now, we have focused on another body fluid that is sometimes a bit neglected, the so-called interstitial fluid or skin fluid. And skin fluid is an enormous amount of fluid that you have in your body. In my case, I might have about 6 liters of blood in my body, but I certainly have 12 to 14 liters of skin fluid, skin and muscle fluid, and so-called interstitial fluid that is really around in all parts of your body, but specifically in skin, in layers below the stratum corneum of your skin. And you may have seen this fluid after having a shallow scratch, or after walking for a long time in shoes that are not well fitting. So you see it in blisters. And indeed, it's a very simple matrix. It contains water, iron, some protein, albumin, some glucose, and some phosphate. So it's actually much simpler than blood. And we have analyzed that and found that this might be a suitable target for a glucose measurement, because skin fluid is containing about 85 to 95% of the glucose that blood contains. And if you select the suitable spot that is well circulated and well capillarized, you may have a very fast adaptation of skin fluid to blood glucose. Now, how do you measure that? Infrared spectroscopy has the problem that it doesn't penetrate very deep into water containing layers. So for example, the penetration depth for 10 micrometer infrared radiation in skin is around 60 to 100 micrometers. No chance for a transmission measurement. The only way of measuring glucose in deeper layers here is either a photothermal or a photoacoustic technology. The typical idea of ATR, attenuated total reflection, for the infrared doesn't work because the penetration depth is much too small, and you stay in the stratum corneum, which doesn't have a relevant glucose content. Some techniques like specular reflectance or diffuse reflectance might go a bit deeper, but still not into layers that contain a relevant glucose concentration. So what we developed is, we actually focused on the photoacoustic or photothermal effect, basically an old story, as you see here. And it's the production of heat waves or acoustic waves as a result of the deposition of heat after absorption and vibrational relaxation. So you excite in the infrared, the molecules go to a higher vibrational level, and they de-excite rapidly in about 10 to the minus 12 seconds. And you can either measure the heat coming at the surface, or you can measure the sound coming at the surface. Let me focus on the latter. The heat coming to the surface can be measured, and we have developed a specific technology for that, namely a photothermal deflection technology. It uses an internal reflection element from infrared transparent materials. It uses an infrared laser, a so-called quantum cascade laser that comes in from the bottom, enters your skin for about 60 to 100 micrometer depth, reaches glucose containing levels, and this laser is pulsed and at the same time scanned across the absorption range of glucose in the 10 micrometer region. It's scanned continuously or step by step, and whenever a glucose molecule is excited, it gets de-excites and deposits a small amount of heat in the skin layers, and this heat actually migrates to the internal reflection element and creates here a so-called temporary thermal lens. Now, thermal lenses are a well-known phenomenon. If you drive in summer on an asphalt road, you may see some house or some landscape here shifted by a mirage effect, and actually this mirage effect is the thermal lens here, and we probe the thermal lens by sending in a second laser beam, a simple red laser diode, and measuring the deflection of this probing beam by a simple position sensitive photodiode. This deflection is proportional to the absorbed radiated visible light. This technology has been developed by us and patented by us, and it's now part of the company's assets, and can be used to measure glucose very precisely. You see here actually a view of our DiaMonTech base model. It's containing the quantum cascade laser, it's containing power supplies, laser control, a detection module consisting of the probing laser, consisting of a position sensitive detection element, data recording and storage. It actually communicates with your mobile phone. And this base model has a RFID module for user identification. So it's a multi-user device, maybe for the use in clinics. It has a learning mode where the device learns with the help of artificial intelligence about the skin of the user, and has a measuring mode that uses this information to for each individual user to measure glucose. Now, why does that work? Why does that work, and why are we convinced that this technology is superior to many other non-invasive glucose measuring systems that have been attempted or are around? None of them is really commercially very strong, but why do we believe it works? First of all, yes, it works in the interstitial fluid of skin with a relevant glucose concentration. And second, it uses a very selective fingerprint of the glucose molecule. So it's very specific and not prone to other molecules' influences here. It uses quantum cascade lasers. It uses a patented photothermal detection that I described to you. And it has the specificity for glucose due to this fingerprint in the mid-infrared. So somewhere in 10 microns, we are measuring glucose. Now, after having developed demonstrators and a first series of prototypes, we have gone to the validation of this technology, and this is usually validated by oral glucose tolerance or oral glucose correlation tests. And I come in a minute, I come to our study 100 with 100 volunteers, but I'm not allowed to show you the personal data of volunteers. So it's actually my personal data. You see me here with the hand on a laboratory setup here. This finger is measuring glucose non-invasively. The other finger is pricked regularly to have a control measurement. And then glucose is used to modulate my blood glucose. So the oral uptake of glucose, usually you select the standard test, which is 75 grams of glucose in a glass of water. It's horribly sweet. You don't like it. It's much worse than the pricking, by the way. If I'm working as the guinea pig in our test, I usually take 100 grams of assorted near-egg or marzipan in less than 5 minutes. That's my preferred one. What you see here is that the invasive and the non-invasive measurements taken at the same time in a distance of 5 minutes over the 2 hours follow very closely to each other. And what you see here is the plotting of the reference concentration taken with a clinical glucometer with respect to the infrared measured glucose content. And you see correlation is pretty good. All these are in the zones A of the so-called correlation grid. So it seems to work. Now, if we take a larger amount of volunteers, this is what we call the study 100, test with healthy and diabetic volunteers, performed about 2 years to 1 year ago. We had 41 diabetes patients, type 1, type 2. We had about 59 healthy volunteers. This is all approved by the Ethics Commission. We had a nice age range. You're not allowed to go below 18 because there's are special regulations by the Ethics Commission. We had a balance between male and female. It was supervised by experts, and we measured in 5-minute intervals over 2 to 3 hours. And the cluster of about 2,000 data pairs, reference invasive versus infrared non-invasive, correlates nicely. Actually, we are at a position which corresponds to commercially available minimal invasive glucometers. We're quite happy the technology seems to work. And at present, we are developing that into different devices. The base model, which has been used for the test, is a shoebox-sized device that we built in a small series. It's actually used to conduct human trials, to go into clinics, to go to diabetologists, and it's CE approved. It has been CE approved early in 2019. The present development goes to a deep pocket model, which is basically the size of a smartphone and allows the diabetes patient to carry it around and to put the finger on it whenever you want, and determine your glucose very efficiently. This is almost complete. The laser system has been miniaturized with in collaboration with laser manufacturing companies. The electronics is complete. The programs, the software is complete, and we're now testing that. And we think that we can get it on the market in mid-2021. Actually, to pick up the arguments of Ralph half an hour ago, yes, we are aware of the problem of manufacturing readiness. So we have a manufacturing partner that is involved in a very early stage already with the design of the electronics and of the mechanical and optical parts. And this device is already a big help for the diabetes patients. From there, we might move on, or we will move on to a band model, which is basically a wearable. For that, we have a very big industry partner already committed to the project. I'm not supposed to name the partner, but it's someone that is involved already in wearables, and it will contain the entire system and allow a continuous measurement of the glucose of diabetes patients. Now, let me summarize that.
Very briefly, Werner.
Very briefly, let me summarize that. I have actually, here we are. We have a system that allows to measure glucose without blood, without pain, and without any discomfort. It will allow more frequent measurements, thus a better control of blood glucose, less complications for diabetes patients, less secondary disorders. There will be some cost efficiency because there are no test strips and no consumables. Overall, we think we can provide a better life for diabetes patients. Thank you.
Thanks a lot, Werner. This was great. I didn't interrupt you earlier to say that you were a bit late because with the marzipan, you made me laugh. So I thought I can maybe yes, further jokes to tell.
Francesca, if you come to Berlin, you get marzipan as much as you want.
I am in Hamburg, Werner, so I'm closer to Lübeck than you.
Yes. Okay.
I know very well what you were eating there, so that's really nice. So I see we have a questions from, no, maybe we don't. No, this is not in the chat, sorry, but I have definitely a question about resolution, because I see the miniaturization was my questions first, but then you had the slide about it, and it looks like you have all the partners already around to go to this smaller one and then to the wearable even, because there is this big guy that we cannot mention, right? Right. So you don't need further support to miniaturization, apparently.
We welcome every cooperation that goes towards miniaturization. At present, we have very close contacts to two laser manufacturers, because what they had to do is to shrink down the sizes of the quantum cascade lasers that have been either boxes before, or the smallest one was a matchbox size, and they shrink it down to about the size of my fingernail. We actually went from a continuously tunable quantum cascade laser to a laser array with 10 to 15 relevant wavelengths, which is much smaller and has much less power consumption and is much more stable. And that's actually entering the smartphone-sized pocket device. That looks pretty good. The precision will be about the same as the system that we have in the box right now. We don't market the box. The box is a small series that we provide for clinics and actually for further validation. And we're learning a lot at the box for new quantum cascade lasers, for new optics. So the box 2.0 is around and is being tested by diabetologists with great success. Now, the band system needs really miniaturization experts, and that's why we have that collaboration with a big company that I'm not yet allowed to call the name.
Okay, that's good. But then you're well prepared in case some bad surprise comes or anything that you might need, you know, that EPIC can help you. And for sure also MEDPHAB, I'm sure if you have some manufacturing readiness level issue, you can ask us.
We are pretty much on the diagonal between MRL and TRL.
Very good. Then it's the perfect spot, sweet spot.
Okay.
Very good. Thanks a lot, Werner. This was really great. So for any connections, any support, we will contact you. If all these quantum cascade laser manufacturers are listening to you, maybe they will give you the third option. Who knows? We will see.
Okay.
Very good. So now we go on to the second page of our agenda. You all can see, so second slot is time for Aldas Juronis from EKSPLA in Vilnius, Lithuania. Welcome. And today we will hear directly from Lithuania, a topic of using of photoacoustic imaging solutions in early stage breast cancer detection. So Aldas, the floor is yours.
Hello, Francesca.
Hello, Aldas.
Yeah, I hope you can hear me.
Hear you and see you. Now we just missed the slides.
Okay, just a minute.
They are coming. Very good. The floor is yours.
Okay, perfect. So, hi everyone. My name is Aldas Juronis. I'm head of OEM Lasers program at EKSPLA. Today I will talk about photoacoustic imaging solutions in early stage breast cancer detection. Okay, in the year of 2018, according to the World Health Organization, a cancer caused about close to 10 million deaths. Globally, about 1 in 6 deaths is due to cancer. 3 most common cancers are lung, breast, and colorectal. Breast cancer is the most commonly occurring cancer in women and the second most common cancer overall. There were over 2 million new breast cancer cases in 2018. Breast cancer alone caused more than 600,000 deaths in the year 2018. Early breast cancer detection and accurate diagnosis are crucial for optimizing survival chances with appropriate therapy. There are a wide range of conventional technologies for early diagnostics, such as X-ray, mammoscopy, MRI, ultrasound, and others. Each one has its own advantages and, unfortunately, disadvantages. For instance, X-ray mammography provides sufficient spatial resolution while maintaining comparatively low cost and simple infrastructure. But it employs ionizing radiation, has lower detection sensitivity in high-density breasts, and thus require often painful breast compression. MRI does not use ionizing radiation, has high resolutions, and possesses high sensitivity. However, it requires the administration of contrast contrast agents, does not have a high specificity, is expensive, and examination takes quite a long time compared to other methods. Meanwhile, ultrasound is safe, comparatively easy to use, but lacks specificity and has the drawback of being operator dependent. X-ray mammography is the only primarily technique currently found sensitive enough for screening. Unfortunately, it has a low positive prediction value. Therefore, other methods such as MRI and ultrasound are mainly being used only as additional diagnostic trajectories. To overcome drawbacks of conventional breast cancer diagnostic methods, brand new technologies are being developed. One of these is photoacoustic imaging. Photoacoustic imaging is one of the fastest growing research areas of non-invasive, high resolution and high contrast visualization of both superficial and deep tissues. Photoacoustic imaging works while illuminating the breast tissue with near-infrared pulsed laser light. Optical energy absorption at specific locations leads to a local pressure rise due to the effect of the thermoelastic expansion. The pressure transient propagates as ultrasound, which can be measured outside the tissues with ultrasound transducers. Hemoglobin as an absorber provides functional contrast to allow visualization of vascularization associated with cancer progression. The production of new blood vessels is called angiogenesis. Inducing angiogenesis is thought to be one of the biological capabilities of tumors and a hallmark of cancer. A quantitative photoacoustic reconstruction algorithm from the data obtained by ultrasound transducers can reconstruct a complete 3D blood vessel system, including tumor angiogenesis. In order to make significant impact in breast cancer diagnosis and to develop, validate, and begin exploitation of a dedicated breast imaging device, a PEARlabs project was initiated. PEARlabs brings together applied physicists, technology developers, mathematicians, algorithm developers, ultrasound detection experts, laser specialists, epidemiologists, and radiologists to work on a new generation system for imaging the breast using both photoacoustics and ultrasound. State-of-the-art near-infrared tunable wavelength source developed at EKSPLA is being used in PEARlabs project. A new generation photoacoustic mammograph that is being developed during this project combines non-invasive 3D photoacoustic imaging and ultrasound imaging. The device provides near real-time, full breast multimodal images to the radiologist. Quantitative spectroscopic photoacoustic images are extracted offline, providing to the radiologist information relating to tumor physiology and function such as angiogenesis and hypoxia. During examination, the patient lies face down on an ergonomically designed investigation table with integrated hemispherical water-filled imaging tank, in which the breast is immobilized without any compression. In this picture, you can see a close view of imaging tank that is being used in the latest a prototype of photoacoustic mammograph. In this picture, you can see graphical view of imaging tank intersection. Pulsed laser light is delivered via fiber bundle with one input at the laser end and 40 separate fiber outputs at the, that are evenly distributed across the imaging valve. You can see these darker dots where the fiber outputs are projected to the bulb. Imaging bulb also comprised ultrasound transmitters for performing ultrasound computed tomography, as well as an array of 512 ultrasound detectors optimized for high sensitivity, with a wide frequency bandwidth and a wide angular acceptance angle. The array measures conventional ultrasound signals as well as laser-induced ultrasound signals from the breast. A quantitative photoacoustic reconstruction algorithm is being used to extract 3D tissue oxygen saturation map from acquired data sets. From the ultrasound mode, the radiologist can visualize anatomical feature and extent of tumors, and from the multi-wavelength photoacoustic mode, he can observe a tumor vascularity. Imaging bowl, tunable wavelength laser system, as well as other components are integrated into ergonomically designed investigation table, which you can see on this picture. On this picture, you can see already third generation prototype of photoacoustic mammograph, which was built by one of the PEARlabs project partners, Dutch company PA Imaging. Such breast imaging approach allows to reduce discomfort for patients that is usually takes place during examination with traditional X-ray mammograph because of a painful pressure applied to the breast. While using the photoacoustic mammograph, as you can see, the breast isn't applied with any pressure at all. The clinical prototype of PAM3 that you've seen in the previous slide has been developed and installed in the Medish Spectrum 20 hospital for clinical trials just recently. Although images from the latest prototype are not yet available for demonstration, here I will present some images that were obtained during clinical trials with the previous generation of photoacoustic mammograph called PAM2. These pictures that you can see on this particular slide were obtained during photoacoustic mammoscopy screening of 78-year-old patient. In the left breast, an abnormality was detected, which you can see circled in each picture. During further histopathological investigation of the biopsy, specimens revealed an intraductal papillary carcinoma, also atypical. Here in the corner, you can also see the parameters for the photoacoustic imaging that were obtained while making these pictures. And as you can see, the resolution of 350 nanometers, while the depth obtained was 4.5 centimeters. And as you can see from this also data that the total examination took less than 3 minutes. This is per one breast. On these pictures, you can see a breast of 49-year-old volunteer. This is without any changes. So this is the pictures of the healthy breast, and as you can see, it's very clearly, you can see all the vascularity, and this is pictures just from the photoacoustic imaging. So main advantages of the photoacoustic mammography that it is a patient friendly, as it doesn't use any radiation, doesn't use any contrast agent, and it doesn't cause any pain during the examination to the patient. Examination is neither hampered by tissue density, nor by the female hormone cycle, nor by implants. In general, this means that this examination, photoacoustic mammography, is suitable for all ages women. Better imaging specificity allows to significantly reduce false positive results. No restriction for time intervals between screenings. This is because no dangerous irradiation is being used for the photoacoustic imaging. And examination takes less than 3 minutes, which is one of the best comparing to currently used conventional methodology. EKSPLA as a company was invited to join PEARlabs Partner Consortium because we have extensive experience building tunable wavelength lasers. Among those lasers, we have a PhotoSonus series dedicated specifically for photoacoustic imaging. Flash lamp pumped PhotoSonus laser, which you can see on the left-hand side picture, is all-in-one integrated laser system into a mobile card. Actually, inside this card, it is a pump laser, OPO, power supply unit, and chiller integrated in this quite small unit. It can have up to 250 millijoules of pulse energy, a wide range, wavelength tuning range, and repetition rate up to 20 Hertz. Particularly for the PEARlabs project, we have developed a customized version of PhotoSonus laser, which has up to 500 millijoules at its maximum of pulse energy. Diode pump PhotoSonus 6, which you can see on the right-hand side, has a high repetition rate of up to 100 Hertz and comparatively high maximum pulse energy for diode pumped OPR lasers of up to 90 millijoules. This laser also has a versatile synchronization and control capability, which enables easy integration of this laser with any photoacoustic equipment. All PhotoSonus lasers has fiber coupled outputs that can be customized for a specific customer's fiber bundle and can have fast wavelength switching that allows to change wavelengths in any order between two consecutive laser shots. Also, PhotoSonus lasers prove to be reliable and does not require any adjustment after long-time operation or after laser transportation. So thanks a lot for your attention, and I would be happy to answer your questions.
Thanks a lot, Aldas. That was a good strategy. You start with what you want to do with your laser, and then you show the laser. So everybody's looking at how was the laser actually. If you do other way around, usually people are then, okay, now it's just the laser.
It's just advertisement. I don't care about that. Yeah.
Yeah, no, of course not. No, but it was a good strategy because now, especially with this audience, we know that there's the lasers that you can apply for this technology. And actually, it was really interesting also to see what you are doing in this European funded project. So basically, you were mentioning that you are now at the third version of the prototype, right? So
yes, it is.
And you showed only because you were allowed only to show images from the second version, right?
The thing is that according to the timeline, the clinical trials of the third generation of prototype was planned to start, if I'm not mistaken, in the very early of this summer. But unfortunately, as we all know, due to the virus, hospitals were closed for such type of trials. Of course. Therefore, it was postponed, and the initial trials of the third generation of this equipment was started only just a few weeks ago. And we are just starting obtaining new images, which are, of course, need to be work on it. They need to be optimized because in parallel, engineers are working on the reconstruction algorithms in order to make perfect pictures. But the main thing is the main difference from the previous version of the prototype is that this one, third generation photoacoustic mammograph, it combines two ways of detection of images. It's a traditional 3D ultrasound tomography and photoacoustic tomography. Okay. So it's not just one, but it combines the best benefits of both these methodologies.
Okay, that means that this will be also the focus of the European project, is also to deliver this device with these two integrated technologies.
Exactly, and it's not just to develop some technology, but also commercialize. It means that we need to prepare the final product, which could be offered to other hospitals for using.
Okay, yeah, that was exactly what I was about to ask. So then basically your duty, so your task is over when you deliver this, and then you try to test them by different potential customers, and then they will decide if they want to buy it or not, of course, and then is the role of this project. Yeah, it is really nice, really interesting. And in your case, for your laser in specific, you said you made a customized product. So that was, what was the difference actually? So you increase the energy per pulse, and
it's double energy. And to our knowledge, there are no in the market, there are no available laser systems that could have such a combined high pulse energy, and at the same time there would be a tunable wavelength.
Okay, and this is only for the photoacoustic part of the technology.
Yes, it is.
Okay.
So of course, these lasers can be used also for other, let's say, microscopy. But we are focusing with this particular for the photosonus series for the photoacoustic applications.
Very good. Very good, very interesting. Thanks a lot, Aldas, for this overview. Also we look forward to see also the third generation that hopefully will be tested soon. We understand, of course, now everything that is happening in hospitals is not research or testing prototypes, because if doctors are busy with other things, it's clear that some delays is included, but hopefully it's getting better. And so also the third generation now is getting all the data and for the new proof of principle of this last prototype. Very good. Thanks a lot, Aldas. And now we go. Yeah, thank you. And then we go on with the agenda that I will show you now quickly. So now we are the third speaker of this slide. So Christoph Schnell, thanks a lot for being here from FISBA in Switzerland. So now he will present to us FISBA Ready Beam, the compact multicolor laser source. Thanks a lot, Christoph. I stop sharing so that the floor is yours. You're muted. You're muted. I see you're talking.
Hello, can you hear me now?
Now, yes.
Yes, thank you very much for having me here. It's a big pleasure to have the chance speaking here. Do you see my screen now?
Yes, if you go to presentation mode.
Yes, I will do so.
Perfect. Thank you, Christoph.
Yeah, thank you very much again. Just some words about myself. It's my first time here, so most probably you will not know who I am and what I'm doing. So my name is Christoph. I'm working here in the role of a product manager for the company FISBA, and I will give a brief introduction to the company with special focus on photonic systems and laser modules. So here we are in the beautiful area of eastern Switzerland, which you can actually really call as an optic cluster here. We are located in St. Gallen. We're here since 1957, and we are a privately owned company still today. Roughly 200 employees with around 30 apprentices. Subsidiaries in Germany and the US, and since 2020, so since this year, we have a sales distribution in China as well. You can see here a picture of our new production building. What means new? It's already 8 years old, with an inside view here below. So that shows you already that, we have our own production here, and we have our own production here in Switzerland exclusively. Some facts about the company within some keywords. Since, let's say, the very early times already, FISBA has been very closely engaged in doing micro-optics for endoscopy, or let's say medical applications in general. And that's a relationship that started that early and that still lasts until to the present day. And yes, we are very proud of extending that relationship. continuously, and still, yes, playing a leading role in micro optics for medical applications since then. FISBA has also been an early adopter of new technologies, if you want. I think you can call that. Computers in 1982, a new technology. So we applied computer-aided design since then, so very, very early. I have been told that the PC that was purchased costed a fortune, and most impossible nowadays to imagine the amount of money that you had to spend for computers that you can now buy for several hundred euros. FISBA used to produce its own interferometric systems. That's a business that was finally sold to the company TriOptics in Berlin days later. And FISBA, and that's most probably not that well known, already started quite early within the laser business. So the first lasers came out by the time of 1993 already, and those lasers were diode lasers for material applications or industrial applications primarily. Yes, as a, let's say, some years later, the idea came out to produce the beam shaping optics for our own lasers by ourselves, and in 2004 finally it was the start of our fast axis collimating lens product line. A very successful product since then, and of course nowadays we do not only use it for our own laser modules, but also sell these FSCs let's say in many different types as a standard product all over the world. If you want, call it a restart, then we can maybe define the year 2013 like a restart of our laser systems. So from that time on, we started again our own development of diode lasers with a focus of laser combiners, and that finally led to the introduction of the FISBA Ready Beam, which came out, or which is in the process of being launched actually this year. That's just another visualization about, I would really say, the very broad ranges of product that FISBA can offer. It's about miniaturization in many cases. So FISBA is really capable of doing very, very tiny lenses. We're speaking about sub-millimeter diameter lenses in that case, with high optical quality, but also miniaturization in terms of micro vision systems, micro cameras, or micro laser modules. I will not mention all the other things that FISBA does, because we are very much involved in customization and customer specific projects, but those include topics like machine visions or modules for the printing industry. Coming back to the lasers, and you see here three photos that, let's say, show maybe three evolutionary steps that mark our restart of the laser business by 2010, and it finally resulted in the FISBA RG beam in the year 2013, which we call a FISBA product. It's still a available product. You can find it on our website. So I like that photo much. Obviously, something went wrong here on the left-hand side with the anodization process. So the material rather turned out to become brown rather than black. The FISBA RG beam, which you can see here, is available in 2 types. We have a free beam option and a single mode fiber coupled option. And you see here, it's very compact, it's very tiny. The specification you can see here on the left hand side. So it's a true RGB diode based system, and yeah, we over the years have sold many in different fields, although as we found out, so there were more applications in the industrial related fields rather than in the medtech area. So this is just an overview about our application areas, or about our market fields, where we are active and where we are selling to. And indeed, we have applications and customers in almost all these fields. As I said, with respect to the RG beam, we basically talk about industrial applications, probably in metrology or in protection applications. Technical endoscopy is rather another niche. If we look at medical applications, then it's rather not the RG beam that we use here, but here we talk mainly about really custom specific laser modules that we produce especially for dermatology, but also for ophthalmology applications. So unfortunately, I'm not able to show examples for those cases, as for so many custom specific projects, in most cases we are not allowed to share those application data or applicational results from them. So that's why I'm so happy that later on in that short presentation, I'm able to share some results that were obtained with our new laser modules later on. Yes, still, there was a part of the market that we didn't address that much with the RG beam, and also not with the customized laser module so far, and that's actually the market what you will call basically the life science market, everything about scientific instrumentation, microscopy, spectroscopy, and so on. And that's definitely one, that was one driver for the development of the ready beam. Before I show the ready beam in the next slide, just something that maybe summarizes why FISBA is really convinced about the concept of a true integrated multicolor lasers. So this is 3 or more diodes coupled in one fiber, or having a free beam output. I think the reduction in complexity more or less directly translates into operational simplicity. So what we want to do with our laser modules, we want people to integrate them fast into their instrumentation and make life easier for them. So I don't know if, maybe most of you, at least the laser expert, they know how troublesome it can be if you want to couple different laser lines into one single fiber, or at one special point where you want to have them for your applications. So it's really, really much easier if you have all that integrated and pre-aligned. In the end, we talk, of course, about time and cost-saving issues. So the necessary next step in that way was the development of the ReadyBeam. It's a very new product, and yes, it's a product which is which is not that let's say typical for FISBA, as I said, but we are a customer project-driven company with some products, let's say, spinning off from those projects from time to time. But this is such a product. It's a new product. What we added compared to the RGBeam that I introduced before is a full electronic driver integration, as well as Peltier tech cooling unit. So yeah, it's more or less a plug and play device that is of course single mode fiber coupled. It can be modulated, and it's available in different diode combinations, and it's a still growing family, and we are also open for input and customer requests in changing and adapting diode combinations within that platform, within that housing. Here are some examples of where the ReadyBeam is currently used or can be used generally. So this is microscopic images that was friendly provided by the Dutch company Confocal Point NL. So they have a very interesting technology in upgrading, let's say, a standard wide field microscope with super resolution capabilities. That's all possible with an external unit. So of course, feel free to visit them and learn more about their technique. What I'm happy about is, of course, that we are allowed to share those images, which the ReadyBeam was used for as an excitation source. And you can really see here in the image of those chromosomes here, and the scale bar. We are here with 200 nanometers, and if you see the spacing between those chromosomes, which are clearly below these 200 nanometers, that we have a super resolution image here, indeed. Another example that I will show in the next slide comes from a light sheet microscope. That's a microscopic technique that might not be as widely known as confocal microscopy, at least among the general public, let's say, although the origins are already quite old. Special thing about light sheet microscopy is that it allows to illuminate let's say large samples, for microscopic large samples of several millimeters within a short time, and produce a 3D rendering out of them. So there's a small light sheet with the illuminated at that MEDPHAB, and the excitation geometry is here perpendicular. These are some examples from a light sheet microscope, and here I refer to Dr. Fabian Voigt at the University of Zurich in Irchel. He is working, or was working, at the lab of Professor Hämchen, and he, let's say, founded the let's say the organization mesospin.org, which has a goal to build up its own and compact light sheet microscopes. Please feel free also to visit that site.
Okay, so is it you, because we would have still 1 minute? Do you want to see again another slide, or?
Okay, I'm running out of time. No, I'm just go quickly. So there's another thing that I just want to show quickly. So it's related, but we are talking here about white light sources. Something that I would like to have to shown directly on the exhibition. So FISBA has produced three functional prototype based on different technologies on a laser activated phosphor. We have functional prototype of a LED combiner, and we have a very compact module based on the single high power white LED. Yes, application areas for such white light sources are, of course, ever then if you want to bring much effective luminance through a fiber, such as in medical endoscopy, for example. Yes, please feel free to contact us. FISBA has, yes, deep vertical and deep horizontal integration capabilities, and we are open for requests. We are open for customization. And thank you very much for your attention. I'd like to close this presentation with another example of image that was taken with the light sheet microscope, and it shows a cleared mouse embryo, which is 11 millimeters in size. Thank you very much.
Thanks a lot, Christoph. Thanks a lot for this overview and also for this new product that you were introduced to us. And I have to say, even before you asked for possible applications, Fokko was writing already in the chat some ideas for you. So maybe Fokko, do you want to tell to also loud so that everybody can hear it also on YouTube?
Well, the thought that occurred to me was that since you have these wonderful laser sources also broadened in the spectrum for RGB, that looks quite interesting. But you, of course, have a library of lasers also in the near infrared. It would be an interesting thought to combine such things, as you can do more than 3 wavelengths as seen in this picture, to also use a near infrared laser and to have the surgeon visualizing not only the visible, but also the invisible. As I said, the camera can see very well into the near infrared, but integrating laser sources in a very plug and playable way, which I think that you are offering, it might be a very interesting one.
Yes, thank you for that input, and it's true. And of course, we know that there are as many applicational needs as there are customers, and we will never be able to cover them all with a three diode laser combiner. So, as I said, we are working on new concepts. We are working on a more modular concept in the lasing area, and besides that, of course, we are producing customized modules which have already more than three wavelengths. That's true. Yes.
Okay, because I can give you one tip there. It's not a secret. It's in any publication on fluorescence. Endocyanine green, ICG, is used universally in surgery as quite a common fluorescence, and it can be very well excited by 785 nanometers, so 785 nanometers. So if you would be able to incorporate such a laser into that stuff, I think you'd already be in business with a lot of applications in medicine. That's not a secret. Any person skilled in the art would immediately recognize this, but it might help you to get into contact on that. It would add this functionality quite quickly, because modulation, of course, per individual laser is also possible. So you can do a lot of with such a very compact module. So your manufacturing readability level, that is already, again, showing you have a high TRL and a high manufacturing readiness. So, hey, I think the first speaker here did a very good job on highlighting that.
Yes, it's true. So, Christoph, you see, you didn't have many applications that you could show in the medical application, but you have now a long way to go, and to just add one wavelength, and then it's done.
Yes.
Thanks a lot, Fokko, for the inputs. I guess
Thank you.
Christoph and FISBA are really happy to hear this, and we are here for this. So very good. If you want more details, you can for sure discuss this also offline. So it's really nice that we can offer this opportunity here with the TechWatch to make this exchange possible, let's say. So thank you, Christoph. Now it's time for crazy. I was like, is maybe I forgot something, but no, it's really the last talk. We are already at the end. So let me show you. We are here at our second Thomas of the day. So Dr. Thomas Bocher from Physik Instruments. So we go back to Germany, to Karlsruhe, and we will hear about precision positioning, a key technology from genome sequencing to digital slide pathology. Thank you very much, Thomas. And the floor is yours.
So thank you, Francesca. Thank you for inviting me. First question is, can you hear me? Can you see me?
I can see you. I can, well, everybody, I assume, and hear you as well.
Okay, so I hope that I can start now.
Yes.
Do you see my presentation?
Yes, if you go in presentation mode, it's better, so we don't see the side. Perfect. The floor is yours, Thomas.
Here we go. So ladies and gentlemen, dear colleagues, welcome to my talk about precision positioning and what it can do for applications as genome sequencing and digital slide pathology. And also welcome to PI. PI stands for Physik Instrumente. My name is Thomas Bocher. My role is Segment Marketing Manager, Microscopy and Life Sciences. This year in 2020, PI has its 50 years anniversary. I think it's worth to quickly look back to a remarkable history of technological innovations and market introductions of unique products in the field of precision positioning. So for example, in 1987, we have introduced the 6-axis parallel kinematic famous hexapod. In 1994, we introduced the famous PIFOC. It's a focusing element for objectives to be used in microscopes. Even today, customers ask our competitors, I need a PIFOC, and they don't know that PIFOC stands for PI focusing element. So we somehow proud that we have even defined a whole, let's say, a product class. In 2001, we introduced very important, the PI line microscope stages using ultrasound piezo motors, very smooth, low noise, low vibrations, low heat. So the best solution for microscopy. Then later on, we introduced several kinds of piezo-based VORC drives. Another one very famous, the Q-motion with inertia drives. And one of the latest introductions are our direct magnetic drives, the PMAG line, which are very fast using the Lorentz force. Now, coming to today's talk. So we are heading for 4 strategic market segments: photonics, life science and microscopy, precision automation and semiconductor. And today, as I said already before, we like to concentrate on 2 cutting edge applications, I think so, in the life science and microscopy field. And this is genome sequencing or whole genome sequencing and digital slide pathology. Now, what is now, let's say, how is it going? How is genome sequencing working, and what role is here precision positioning playing? First, that you get a better understanding how these instruments look like. Here you see one from Illumina and one from BGI. So you see large desktop instruments. They are packed, they're full of microfluidics, lasers for excitation, optics, and precision drives. By the way, these are only example images taken as free license images from Wikipedia, because we are not allowed by NDA to talk about, give the names of our customers. But what I can say is that we are supplying the largest makers of genome sequencers in this market segment. I think it's really stunning when we look at what happened to the price per genome sequencing during the last 20 years, because this illustrates what really happened to this industry and to this technology. At the beginning of this century, in 2001, it was literally 1 $100 million was the cost to sequence one whole human genome. And now in 2020, the biggest two players, Illumina and BGI, they claim that they now can do it for $100. So it's 6 orders of magnitude that the price dropped. And I think this tells a lot that this technology could now play a really big role in our society. And when we now look, okay, what are now the use cases? What are the applications? I think you all know them very well, but I just like to point it out. So the personalized medicine, and we had a good example when Aldas talked about breast cancer, because this is a possibility for women, for example, to find out if they have a high risk to develop breast cancer in their life. And maybe you remember there was a case in the press, the famous actor Angelina Jolie. She did it some years ago and she took a drastic decision afterwards. So this is one, let's say, result of it. But in the end, you can check your whole genome for different diseases you might develop in your life. And you can even choose the right nutrition that fits you the best for your life and things like this. Another, I would say, strongly discussed field is the birth planning. I think all of you have already heard about pre-implantation genetic diagnosis or the prenatal genetic diagnosis. There are a lot of discussions if this should be done and in what cases it should be done. But the technology here delivers the possibility to do it if it is necessary. And this is especially true for the virology. I think here we all agree that it is a great help and that these genome sequencers not only sequence the human genome, but also of viruses. So the dangerous Epstein-Barr virus in 1984 was one of the first ones. And then the SARS virus in 2003, and this year, finally, in January of this year, 2020, the SARS-CoV-2 virus, so the COVID-19 virus, was detected by genome sequencing. And there are even more applications. So those who sometimes look at criminal TV series, they know that you have something like the DNA profiling to find out who is now the murderer. Or other important questions, who is the father of an individual? Here it's interesting to see that you only need to look at 10 to 20 genomic regions within the whole genome of a person to be able to say if this is the child of a person or if this is the father of somebody. So really, I would say interesting. And there are more applications in research and in genotyping. But now, let's have a look how it really works in detail. Just to quickly recap how the, let's say, DNA is structured. Okay, we have the 2 oligo strands and they are connected or interlinked by base pairs. Here the good thing is we only have 4 different DNA bases: adenine, thymine, guanine, cytosine. And they can, because they build pairs, they can only create 4 different base pairs. So the task in the end is to read the sequence of these 4 different base pairs. And 3 base pairs in a row are called a codon and form something that is called the letter of the book of a genome of a person. So now how to read now these base pairs? Please look at the numbers, though there's a series of images now, how it works. So it starts with a flow cell. A flow cell is a cartridge, 10 centimeters to 10 centimeters about in size. And you have thin channels to produce a laminar flow, that you don't get turbulences because you have to flush this flow cell many times during the whole process with chemicals and with liquids. You will see in a moment which ones. So laminar flow. In one of the channels now you see on the image number 2 a magnification. So you see single-stranded oligo strands. To get these, you first have to do the following. You have to, let's say, somehow denaturate a little bit the DNA from its tertiary, quaternary structure, make it single-stranded. You have to do PCR amplification, and you have to fix it with biotin to the bottom of this channel. So you have to do something before. Interesting or important is that these single-stranded oligo strands have, in most cases, there are different methods, but they are rather short to medium chain lengths of 300, 500 base pairs. So they are not too long. You have many of them. And now it comes, the principle itself. Let's say, the core technology is fluorescence microscopy in the end, because what you are doing is you do sequencing by synthesis. That means you go from the uppest position down and you, let's say, complete the base pairs. And this you do by flushing the cells with these bases. Each base is coupled with a certain fluorophore that you can distinguish them. You have 4 different kinds of lasers, 4 different colors. When you now go to the image number 4, you see, for example, here, thymine fluorescence is detected at a certain position in a channel of the flow cell, giving a small color dot. And because you do this highly parallel, so you flush always with all 4 bases, you illuminate with all 4 laser sources, and then you get in one height level, you then get an image with the different light points. And when you then cut off with different chemicals, cut off the light and flush the next time, then the next level, then you get a number of layers, of image layers, and putting them together in a computer later on, also finding out which pieces had been cut into pieces by the shotgun technique. Then you can, let's say, generate the whole genome of a person. So this is how the principle works. And to get these colored image dots, you need optics to scan over the flow cell. These optics have to be in focus, have to be very fast. The flow cell has to be adjusted. And for all these duties, you need precision drives. So the first thing is, when you introduce a new cartridge, the loading process, you have to make sure that the tethered set position, so that, let's say, the orientation to this optical scanning system is correct. And for this, you can, for example, use these 2 solutions from our side, from PI, either a DC motor drive or a 3-phase magnetic drive. Important is that they can carry a certain load. Here, it's typically 0.5 kilo. The repeatability is below 0.1 millirad. So, and important, you need high speed. All of the drives should have very high speed, because it's clear when you have such a low price for one genome sequencing, everything has to go really, really fast and highly parallel. The next thing is the set position, because you are scanning over the flow cell with high speed. So either you do step and settle, or you do a kind of a dithering mode, so fly over mode. But in all cases, you need a very accurate set position. So for this, you can use our famous Piefox. I mentioned them already before. It's a piezo flexure-based technology. It's below 5 nanometer precision. And if you like to be extra fast, you maybe go to a voice coil. So a magnetic drive it's a little bit higher value for the repeatability, but it's absolutely fast, also chosen by our customers. And finally, the XY positioning. Here, the favorites of our customers are, again, the magnetic linear drives, because they have such high speeds. They can speed up to 5 meters per second, so really fast to scan the flow cells. But also piezo motor driven solutions like the U780 or similar systems are used for this application. Now, quickly to see that the second application I have in my list, the digital slide pathology, works, I would say, pretty much the same, but it's typically a bright field microscopy application. So first in histology, again, to detect tumors, or the famous pap smears in gynecology or in hematology to see if you have there tumor cells. And the point here is that latest in the 1990s, there happened a change in how doctors are doing this. before the pathologists manually worked with glass slides and did visual inspection. Here you see on the left side how it works with a second opinion, which is worked at that time, which is, by the way, also necessary nowadays. But you see here on the right-hand side, nowadays, the pathologists, they use digital slide scanners. Here again, an image from Wikipedia, and only look together on monitors. They can be even in remote places. So the principle here works very similar to the genome sequencing. So you go in a meander-like structure, scanning your sample. You do a superimposition of all these images, typically overlap 10 to 20%, and then you create the whole image later on. Finally, and this is my last slide, you come again to drive solutions which are pretty similar to those are used in the genome sequencing. You need typically very fast XY stages to quickly scan the glass slides. And in the set position in your autofocus, you need to be both fast and accurate. Can either do it step and settle or in this dithering mode. So thank you for listening from my side. Thank you.
Thanks a lot, Thomas. You saw now I switched out the camera that was very close to the screen to see all the details. No, it was my fault. I should have switched it on a bit later. No, it's really interesting. Thanks a lot also from my side, especially because I'm not used to see PI in a more medical application. So it's really a nice surprise to have you in this TechWatch. Thanks for presenting this. So I wanted to ask you, especially for the sequencing, but also for the second application, in all the cases you are holding the sample, right? So it's the sample that you are moving, and that the whole device does the sequencing, or in the second, the pathology study.
Yes, in general, yes, but it depends a little bit in this regard. So for the genome sequencing, we definitely hold the flow cell. That's correct, with our TETAZ drive, but the illumination and the optical, let's say, scanning unit is above it. So it's coming from above. So it's holded in a second place, so to say. So it's moving over the flow cell. Sorry, because maybe I have not mentioned this. It's an important point, and it's very similar also with the digital slide scanning. It's about the same. Yes.
Okay, very good, very good. And so then that means that now, be careful. I'm not saying that PI products are expensive, but you showed this incredible slide at the beginning about the genome sequencing drop of price. Yes. So could you see also that your contribution to such a device also in the past years got more cheaper, because fast and precise usually don't go together, but you are managing this. So it sounds like a super challenge that you are like winning with.
Yes, you are very good at analytics. You got the point. It's true, because when you go down such extreme way, this means that there is a lot of pressure on the suppliers from the system integrators, and this also happened to us. But you see, I'm still talking about it. We are still in the business, but it is true. They ask us more or less every year to make it cheaper and faster. By the way, faster and cheaper. That's what you have to do. You know, such is life, but we are still here and still laughing. So
yeah, that's very good that you took it with the right approach. So, and that's, you need this input from outside to get always better and also always more affordable. Yes. So very good that is someone outside that is pushing you towards even better results than what you have already. And so really thanks a lot, Thomas, because we know PI is good.
Thank you. So, thank you.
So now I guess it's time for send you to the break. So I don't want to steal you too much time from your free time till the next session. So I would really like to thank you, all the speakers that were here, for nice also discussions, for nice interactions, future plan of cooperation. So it was really nice on the epic atmosphere. I really thank you also who was watching us in the Zoom, in the alternative webinar Zoom room, and also on YouTube, now live and also soon recorded. So you will be informed when this recording will be ready and then posted online, so that you can watch it in the case you were not here today exactly on Tuesday morning. So thanks a lot also IVAM for the nice organization of this and working together. And I guess you are ready for the break, and see you next time, also with EPIC and IVAM in a few minutes after the break. Bye-bye.
Bye-bye. Thank
Thanks,
you.
Francesca. Bye.